thanks so much for having me. I'm really excited to talk to you today about some of my work looking at eco-evolutionary context and how it influences a plant invader's response to climate. This work, as I'll mention, is a really big collaborative project. So I have an et al. Here for all the authors, but in the next few slides, you'll see that this was a huge collaborative effort that required, on the order of 50 to 100 extra hands to get this work done. Okay, so a little bit about myself and how I think about ecology and evolution broadly, and then in the context of invasive species, which is obviously really important to you all. I investigate how evolutionary and ecological processes interact and feedback on each other in the context of climate change. And I've been thinking about this for a long time, and in my lab, I really focus on trying to connect historically disparate fields of evolutionary biology and things like population, community, and ecosystem ecology. So I have an example of an ecoevolutionary feedback here that's relevant to carbon cycling, but we can think about how processes such as selection and evolution, if they're on traits that are important for community or ecosystem level processes, might add up to really important interactions and feedbacks between these fields. So as Kayla mentioned, I use experiments coupled with modeling to really understand how plants and historically some other organisms respond to climate change and the implications for those responses. So I kind of got my start, and I was so happy to see that Mike Kuntz logged on because he is also from the tribolium world, and so I'm glad I had this slide here. And I think I actually stole a couple of his pictures that I didn't cite him for. So these are Mike Kuntz's pictures, they're really good. When I was at Colorado State University in the same lab as Mike, I was really interested in big questions about how evolution can influence ecological processes. And I answer and address similar questions today, but in different systems. So today I'll be talking about my work in cheatgrass, which is an invasive plant that is really abundant in the Intermountain West. But a lot of my work is in coastal marshes and not with invasive species, but really dominant species that shape ecosystem processes, but also asking questions about how rapid evolution of these plants can influence ecological dynamics. So although I use different systems, I'm really motivated by big questions that bring together the disparate fields or historically disparate fields of ecology and evolutionary biology. As I mentioned, this is a big team effort. So here are all the co-authors of this talk, and a paper that the results I'll talk about today is currently in review at New Phytologist. I bolded here some of the leaders of this project, and they're also highlighted in the photos. So big thanks to all the folks that contributed to this. And as you'll see from the extent of the experimentation that we did, and really mostly other people did, and I had the opportunity to analyze the data for, this wouldn't have been possible without such a big team. So who are these people? These people are part of a consortium called Bromecast that was put together as part of a National Science Foundation grant. And in the proposal, it was written that our goal is to learn how to predict the effect of climate change on the population dynamics of cheatgrass or Bromus tectorum across its range in Western North America. And if you're not familiar with cheatgrass, it's really a pervasive and damaging invader in the Intermountain West. And this map on the left here is showing some, distribution and cover of cheatgrass as of 2017, where the dark spaces represent where cheatgrass is really dominant. And so you can see across really big swaths of land, we have quite a lot of cheatgrass. And cheatgrass came over in the late 1800s, probably by accident, and it has been able to, in the course of decades to centuries, has really dominated much of the West. It's also on the East Coast, but it doesn't, for some reason, seem to reach nearly as high densities and have such a damaging effect. Our group uses reaction norms, or this idea of how phenotype varies as a function of an abiotic driver, as a framework for thinking about how we can make these predictions of how cheatgrass regional dynamics will change as a function of climate change. And so if you're not familiar with reaction norms, this figure kind of orients, so what kind of information do we need to build such a reaction norm to make these predictions? Well, we need information of, where cheatgrass populations are and maybe some of the genetic information of those populations. We need information about their phenotypes or their traits, maybe some things that are related to their physiology or phenology or timing. We want to know things not only about their traits in isolation, but how these traits might shift in response to intraspecific and interspecific competition. And using all of that information together, we can hopefully make some specific predictions about how different vital rates like survival, fecundity, or fitness might shift as a function of an abiotic driver, where each line here on the bottom right represents a unique genotype. So each genotype or each source population might have different responses to abiotic drivers over time and space. So why would we want to study cheatgrass? I mentioned that it is a damaging species, and I'll get to kind of the ecological consequences of a cheatgrass invasion in a second. But it's also a really tractable system for trying to make these kind of predictions. So there's both a lot known about cheatgrass invasion and then still a lot of really important unanswered questions. So it's a really tractable system for characterizing genetic diversity because it's a diploid grass that is primarily selfing. It's an annual. It lends itself well to big experiments. We can work with inbred lines to allow for replication of near clonal genotypes. But there's still important questions that kind of preclude us from making really robust predictions about how it's going to spread, particularly in an uncertain climate regime. So I'm showing here some results of some of the important genetic variation and distribution. And I just want to note that I won't be able to talk about a lot of this today, but Diana Gamba has a paper that's in press from our group at Nature Communications, so it should be out any day now, that goes over a lot of the genomic variation across both the invasive and native range. So be on the lookout for that. Another reason that we are concerned about cheatgrass is not only from like an interesting ecoevolutionary, basic ecology question, but also from a really important management perspective. So cheatgrass really alters the environment that it lives in. So not only does it just have a lot of cover across the big landscape, but it alters the fire cycle in these landscapes. So whereas historically these areas have burned on the order of every decade or so. They're now burning every few years. And not only are they causing, these burns because they flower early, they produce seed early, and they produce a lot of fuel for a fire, but they're also highly disturbance adapted. So they're also the first thing to establish following such a fire. So this kind of positive feedback for cheatgrass creates this environment where we used to have kind of these really nice native sagebrush ecosystems that have transitioned into kind of monocultures of cheatgrass. So the main question that I'm going to address in today's talk is how can we predict plant fitness, or you can think population growth rate, as a function of climate? And really, I want to note that I'm not going to be doing any real predicting here. I'm mostly focusing on inference. I'm not going to be doing any kind of out of sample predictions, but that is a big part of our project and is the focus of a postdoc that I am mentoring right now, Becca Nelson. And we are really getting into the nitty gritty of her analyses, and kind of a sneak peek to that is we might know a lot in inference, but we might still be really bad with predictions. So we'll kind of see how that additionally pans out. Okay, so the main question that we want to understand is how fitness or any kind of vital rate of cheatgrass changes in response to an abiotic environment. And on the surface, this seems like it should be a simple answer. We maybe can create a line or some other functional response that would relate something like soil moisture or temperature or precipitation overall to predictions in fitness. But over time, ecologists have found that that is actually quite difficult, not just for cheatgrass, but for a variety of different organisms. And one of the reasons that people think that this might be the case is because some biotic rather than abiotic drivers govern population dynamics. This is a very kind of classic debate in ecology literature, thinking about things like, does the fundamental or realized niche, influence distributions, whether it's abiotic or biotic. But what we would argue is that maybe it's important interactions between the abiotic and biotic environment that provide important context for explaining variation in things like fitness across a landscape or time. So I'm going to present a few different ways that we can think of these abiotic and biotic drivers interacting into kind of separate contexts. So we can think of this in an evolutionary context. So this is really just kind of showing that same reaction norm concept and thinking about genotype by environment interactions. So we might have a bunch of different genotypes of cheatgrass, for example, which we know exhibits a lot of genetic variation, and they might have different responses to the abiotic environment or their fitness might respond differently. And that could create some uncertainty in that relationship between abiotic environment and fitness. If these genotype by environment interactions are structured spatially, this might be indicative of local adaptation. We are going to look at local adaptation in a specific way, given the context of our experiment, where we can think of genotypes that are, if they're grown in different types of environments, if they're grown in a climate or an environment that's very similar to what they're used to, their climate mismatch in this case on the x-axis would be 0. And if they're locally adapted, that means that their fitness is highest when their environment is the closest to what it's used to. If the current environment that it's experiencing is hotter and drier or cooler and wetter than what they're used to, they may see a decrease in fitness. And that would be indicative that they're locally adapted to climate across their range. And then finally, there may also be some important ecological context, in this case, thinking about biotic interactions within species. So how a population responds to a biotic environment might relate to how many competitors or conspecifics are around those individuals. And the relationships of these lines of high density and low density here aren't really specific, but we can think that the slopes of these lines might vary depending on what density that these populations are at. And we could also make this additionally complicated by thinking about how density dependence and local adaptation might interact, where the relationship between climate mismatch and fitness and how quickly you fall off your fitness, depending or based on how far away you are from your climate optimum, might depend on what density you're at. And this is particularly important in the context of invasive species and as they're expanding their range, because they both vary in core and edge populations vary in their density kind of naturally. And whether or not they are locally adapted can tell you something about their propensity to succeed in a novel environment. So to address all of these different types of contexts and how they might influence our understanding about relationships between abiotic drivers and fitness, we conducted a very large replicated common garden experiment with 96 cheatgrass genotypes that were collected across the Western US and British Columbia. Um, Here in panel A on the top left, I'm showing the 4 replicated common garden sites that were in Idaho and Wyoming, Wildcat, Baltzor, Sheep Station, and Cheyenne. You'll notice that Wildcat and Baltzor are geographically kind of right on top of each other, but they vary a lot due to very big differences in elevation. We had different climate that the plants experienced based on the common garden, but we wanted to have a little bit more climatic variation. So we also used a technique of, creating bare soil and then also adding black gravel and white gravel, which you can see in the bottom left, which influences the surface albedo. On average, the difference between the black soil or the black gravel and the white gravel is like a 2 degrees Celsius difference, which can also influence the soil moisture. So I'm showing kind of all of the different environmental all the different kind of combinations of environmental variation across sites. We did this across 2 years, and then we have those 2 different gravel combinations. So the x-axis here is we measured daily soil temperature using probes, and then similarly daily soil moisture and the averages across the growing season. So you can see here that we're kind of spanning a good bit of variation by having these different sites, gravel treatments, and then again across a couple years. I mentioned that we collected genotypes across 96 different sites. I'm showing those here. So we have, quite a bit of variation all the way down to the Mojave Desert and those red plots in Nevada and Colorado. And then up in British Columbia, we've got some really cool and wet source environments. We did a principal components analysis to kind of characterize the source environment from where these genotypes are from, which is shown here in the colors that basically runs from a hot to dry in the redder colors and cool to wet in the bluer colors. We then wanted to capture that information of climate mismatch. So how different is the climate that a replicate of a genotype is experiencing in a common garden environment than where they are from? So basically we did just that. There's a little bit more tweaks mathematically, but the overall process was we characterized the source environment using information of a climate norm where that genotype was from. We had replicates of all the genotypes in the different current environments, and we could kind of subtract the differences in those environments to get some measurement of climate mismatch, where higher values of climate mismatch should lead to decreases in fitness, either positive or negative, if these populations exhibit local adaptation to climate across the introduced range in North America. Oh, I should mention, I missed the densities. The density is here. So we also manipulated densities of the cheatgrass plants, and we did this by manipulating the size of the plots. These bigger rectangular plots have a much lower density, so the cheatgrass plants are further apart, and the square small plots have a much tighter density. Okay. So we were able to individually track over 22,000 plants. Again, I say we, a lot of this was not done by me at all. I want to give big thanks to all of the folks that did that work. It was very painstaking. Here I'm showing a picture of Nikki Pertel, who was a really important tech at Utah State, where I did my postdoc for this work. And it was a huge labor of love. I don't know how much love she would say it was, but it was really hard to get all these data. And but without these data, we would not be able to have the kind of results that we do. We tracked flowering phenology, so when these plants were flowering. This work is already published, and I won't talk about it today. I'll instead focus on whether or not plants survived reproduction, and then if they did survive reproduction, how many seeds did they produce? A lot of these plants produced a number of seeds that is unreasonable for us to count, given any kind of time scheme. So we did subsets and counted and estimated the number of seeds given subsets and inflorescence weight. We then used a hierarchical Bayesian hurdle model to simultaneously understand how our ecoevolutionary context of our current climate, the genotype information, genotype by environment interactions, and that climate mismatch variable influenced both survival and fecundity. And then we were able to calculate fitness as a derived quantity from the model, where fitness is the probability of survival times fecundity. So what we found is that the genotypes that we grew across the common gardens and in these different environmental conditions really varied in their survival and fecundity responses. So first I'm going to show you their variable responses to soil temperature, which again we measured with the soil probe. On the top panel is the probability of survival. Each genotype is shown as an individual line, and they are colored by their source climate. The bottom panel represents fecundity, or the natural log of the seed count. And so we can see general patterns that if you increase soil temperature, you increase the likelihood of survival. And if you increase soil temperature, you decrease the number of seeds that are produced. We are also interested in understanding if source environment or the colors of those lines explained any particular patterns we were seeing in the slopes. And so we calculated the relative sensitivities of each genotype to temperature. So that's what you're looking at the scatter plot on the right. So for example, this top right plot basically takes the linear slope of each of those curves and plots it on the y-axis, and then we plot it against the source climate on the x-axis. So any change in the dashed line would represent that depending on what source environment you're from, you may or may not be more sensitive to soil temperature. So we can see for probability of survival, there's really no pattern, but the source environment doesn't really explain variability in sensitivity to temperature. However, in fecundity, we see that the genotypes from cool, wet source environments experience the greatest declines in fecundity with increasing temperature. So these blue points have, on average, more negative slopes than the other colored or more red colored points. We can look at similar graphs for soil moisture instead this time. So we do see some variability in responses. Overall, we see declines in survival with increased soil moisture and increases in seed count or fecundity with increased soil moisture. But we don't really see that source environment explain sensitivity to soil moisture in the common garden differently than temperature. And because we have survival and fecundity in the same model, we can make some predictions about fitness, which is really the metric of interest. And we see this very interesting flattening of responses, which I'll talk about in a second. So overall, there isn't really a pattern to how soil temperature and soil moisture influence fitness. There isn't like a consistent increase or decrease, and it really depends on what genotype you are. And we see this particular pattern for genotypes from warm and dry environments experience greatest increases in fitness with increasing temperature. Again, we don't really see much variation in response to soil moisture explained by source environment in the bottom. So as I mentioned, this flattening in responses was kind of surprising to us, and it reflected from a trade-off between survival and fecundity responses. So I'm just showing the overall responses here to temperature, the patterns of responses. So generally, we saw an increase in survival with increases in temperature and a decrease in fecundity. We thought this was kind of perplexing, but might represent just an overall plasticity in trying to keep fitness high. So if you can keep fitness high across a wide range of abiotic variables, that might mean trading off between survival and fecundity to keep your fitness response consistent. So we saw some evidence that source environment influences response to current environment, and that is somewhat evidence of local adaptation. But we have this climate mismatch metric to kind of look at that more directly. And we see really strong support for local adaptation to climate. So again, this is that climate mismatch metric on the x-axis, where a value of 0 means a plant was grown in a common garden environment that's very similar to where it's from. And on either side, the environment is hotter and drier or cooler and wetter than what it's used to. We saw this interesting pattern that fitness peaks slightly higher when the current environment is hotter and drier than the source environment, which is not particularly great thinking about how things are projected to change under climate change. We also found some really interesting interactions between our density treatments and local adaptation. So this is that eco-evolutionary context, and although the effect is relatively small, there are significant differences here between our low density, the shape of the curve here, and the high density, where the low density populations or plants, the plants that experience low density have a stronger curve. So as you increase or decrease the environment, you move away from your climate optimum, you see bigger decreases in fitness relative to the high density plants or the plants that were grown at high densities. This could be potentially a good thing when we're thinking about range expansion. So when plants are at the edge of their range, they're at low densities, and that means if they're experiencing a novel environment, that might be difficult for them to do because they're like more strongly locally adapted, or that local adaptation is more evident when they're at low densities. We also wanted to just kind of get an idea of the effect size of these differences. It's a little hard to evaluate with all of those different genotype lines. So we selected a few kind of icon genotypes to take a look at that really varied considerably across our source environment gradient. Joshua Tree, California being a really hot and dry genotype, Summerland, British Columbia being our cool and wet, and then Vernal, Utah just being kind of somewhere in between. And then we made predictions with our model if they were at the coolest and wettest environment or hottest and driest environment. And we saw that our model would predict that the Joshua tree genotype would produce, on average, nearly 4 times more seeds in a hot and dry environment than a cool and wet environment, assuming an average density. And conversely, the Summerlin genotype would produce nearly 3 times more seeds when in a cool and wet environment compared to hot and dry. So these are really big differences in the number of seeds that these plants would produce. And we can think about as genotypes are maybe shifting or moving across a landscape, this could really dynamically change the population dynamics. And finally, although I mentioned at the top of the talk that I wasn't going to make many predictions, and that is really the focus of my postdoc's work, we were somewhat unsatisfied by not making any predictions in this work. So we did do some simulations with our model to think about kind of what we would expect some of these dynamics to be given what we understand. So with the statistical model that we fit, we simulated a 2 degree Celsius increase in temperature across all of our source populations. We assumed that our source populations are currently locally adapted to their source climate, which might be an okay assumption. It might be a little bit unreasonable given that the environment is rapidly changing. And then we assumed average soil moisture and density. Again, maybe not the best assumption. Soil moisture probably changes with changes in temperature, but is a lot more difficult to pinpoint for all of these locations. And then we just predicted what the change in fitness would be for each of these source populations to see if we could see any consistent patterns. And so we found something that is not particularly encouraging, which is that cheatgrass populations seem to be increasing in fitness across many of our source populations. So on the x-axis here is the soil temperature from the 30-year climate normal of all of these source populations. And the y-axis is log and fitness, and each of these arrows represents that change. So the endpoint of the arrow without or the beginning of the arrow, I guess, is what it was before that 2 degree increase in temperature, and where the head of the arrow is after that 2 degree increase in temperature, where the color of the arrow represents if it's an increase or decrease, and the hue of the arrow also represents a magnitude of change. And so we can see here that overwhelmingly there's an increase in fitness, but this is particularly true for genotypes that are in cooler climates. And this kind of makes some good ecological sense. So if you are already in a really hot climate, so you're further along on that x-axis, an additional increase in temperature might not increase your fitness at all. It might actually decrease it a little bit. But if you're in a particularly cold climate, a 2 degree increase could really increase your fitness. In particular, we think the mechanism is here that it could increase your overwintering survival, which can be a limitation in some of these colder climates. So I hope I have convinced you today that accounting for ecoevolutionary context is kind of needed to understand this really important question that ecologists and evolutionary biologists have been trying to answer for decades of how plant populations will respond to an abiotic environment, particularly in the context of climate change. If we can't answer it now, knowing what the abiotic environment is, we we'll still not be able to answer it when the abiotic environment changes. So being able to understand some of the context dependence here, although it's, context dependent is so rampant in the field of ecology, is kind of needed here to really understand this variation in fitness for organisms that are really important to both ecological and economic health, such as cheatgrass. And with that, I'd be happy to take any questions, thanks to funding from National Science Foundation. Thank you.
Thank you so much, Dr. Vahsen. That was a really great talk. Very interesting. We do have one question in the comments, and I'll go ahead and read it. Sarah asked, did you measure soil nitrogen? BRTE responds to nitrogen, which is one of the reasons for such a strong pulse of BRTE after wildfire.
Yeah, that's a great question. We did not measure soil nitrogen, and I think had we gone back, we would have loved to get more information about the soil. We have some soil variables that are going into Becca's analysis, thinking about soil carbon, and then I think we have information like percent clay. We actually might have some nitrogen data, but that would only be for the current environment. We definitely don't have it for all of the different source environments. So yeah, but we have thought a lot about, what we are missing, and particularly what we're missing below ground, which is really important for these plants.
Thank you. If anybody else has a question, I'll unmute you. Just raise your hand, just like Nate did. Okay. There we go.
Thanks. Thanks. That was a great talk. Can you hear me okay? Sorry.
Yep.
I'm bad at Zoom sometimes. Um, I was wondering if you thought at all about the like genomic underlying or like the genomic mechanism that could be explaining some of the like variation that you're seeing in the cold adapted versus the hot and dry adapted. Like if you think it could be driven by like sort of maybe like rapid evolutionary elements like transposable elements or jumping genes, or if that was like different genotypes, like the specific differences in the genetic makeup of them that you think could be driving these patterns.
Yeah, that is a big focus of our project, not one that I have the right expertise to address. So Diana Gamba is kind of the head genomics. She was a postdoc on the project and then now is faculty at New Mexico State. And I think her recent paper that's coming out in Nature Communications might address some of the questions that you have. But we've thought some cool things that we have found is that we see varying selection patterns that have already kind of developed in the introduced range. So in our coolest and our coldest common garden site, we see selection for like a different phenological strategy, like life history strategy than we do in our hot and dry site. Patterns that are like not only a change in the slope, but like a change in like sign. And so we were really surprised to see that, really strong change in selection that has developed over the course of 100 years. So I think these patterns of local adaptation are, yeah, they're like way stronger than we would have imagined. But some of the genomic details I can't give you a great answer on, but I would really recommend checking on that paper.
Cool. Thank you.
Mhm.
Yeah, I'll make sure to, when that paper comes out, I'll be in contact with you, Dr. Vass. And when that paper comes out, I'll make sure to share it in our next newsletter so that everybody
Yeah, that
can
would be great. Thank
see
you.
it. We have another question in the comments. So Morgan had said, really cool work. And I'm curious how you're thinking about the potential trade-off between survival and fecundity, given that cheatgrass is an annual species.
Yes, this one puzzles me. So if other people have ideas, yes, we left it kind of somewhat vague in our in our manuscript in hopes that a reviewer would have a good idea potentially. No, but it's I think the goal rate is that these plants are trying to maintain high fitness and per, like maybe where they're constrained. So at like really low temperatures, they have a hard time surviving. And so can they realize that their population has very few survivors and then produce as many seeds as possible to maintain high fitness? I don't know. Um, I've talked to some other folks that have seen a similar pattern in other annual species, but I need to dig in a little bit more to really understand, like mechanistically, why we'd be seeing this. I mean, we can't exactly ask why, given the context of our experiment, but I think we could do better in coming up with some ideas. So certainly open to others' thoughts.
Great,
Yeah,
thank you.
thanks. I wonder if
Any
density is part of that too.
Hmm.
Yeah.
Yeah, for sure.
any other questions or any other comments relating to this topic? Feel free, if you can unmute yourself, feel free to just ask or raise your hand. All right. Well, if there are no more questions, Dr. Vassen, are there any other comments you'd like to end with? No? Okay.
Yeah, I don't think so. Yeah, thanks so much
Okay.
for having me. It was a really great to share some of this work. We're really excited about this. And yeah, keep your if oh, I do have one announcement. Yeah, if you're a postdoc or a student and you are looking for some data to work with, we are in a too much data, not enough people to analyze it problem. So if this kind of data is exciting to you, feel free to reach out to me or check out our website, Bromcast Network. We are excited to share these data to ask like a lot more questions. But we just don't, the funding has ended. And so but we have a lot more questions that we could ask with it.
Great. Thank you. Well, thank you so much for your time today, and thank you for everyone that attended.